Episode 5 named the wet lab validation bottleneck as the field's hardest constraint. AI generates thousands of candidates, but a lab can validate only tens, and it floated robotics plus a self-driving labs as one response that's still capital-intensive and early. This episode is the deep dive on that response, what a closed-loop lab actually is, why it matters, and why Episode 5 was right to hedge. Let's restate the bottleneck, because it's the whole motivation. Every predictive stage in this series, targets, structures, molecules, ADMET, ends at the exact same wall. A physical experiment has to confirm the prediction, and physical experiments are slow, manual, and rate-limited by human hands and lab schedules. And here's the uncomfortable part, making the prediction faster doesn't move that wall, it just makes it more visible. A self-driving lab is the one approach that attacks the wall directly, by automating the experiment itself. So what is a self-driving, or autonomous, lab? It's a closed loop with no human in the inner cycle. Step 1, the AI proposes the next experiment, which molecule to make, which assay to run, drawing on the design, screening, and ADMET stages from earlier episodes. Step 2, robotics executes it physically, with automated synthesis, liquid handling, and assay platforms. Step 3, instruments measure the result. And Step 4, that result feeds straight back to update the model, which then proposes the next, better experiment, and the loop just repeats, without stopping to wait on a person. The design principle is active learning in the physical world. Don't run every experiment, run the one that most reduces the model's uncertainty. It's the same active learning idea from virtual screening in episode 10, except now the query is a real robot doing real chemistry, instead of a docking calculation. Why is closing the loop the real unlock? Because human-in-the-loop discovery is gated by cycle time. You design something, you wait days for a chemist to make it, you interpret the result, and then you redesign. Closing the loop compresses that whole design-make-test-analyze cycle from weeks down to potentially hours, and, crucially, it lets the model's own predictions be tested and corrected continuously, so it improves on the exact chemistry you care about, rather than on some static training set. This is the most direct attack there is on the Episode 5 triage risk. If you can validate hundreds of candidates instead of tens, you throw away far fewer of the genuinely good ones. But Episode 5 hedged for good reasons, so let's name the real limits. First, capital intensity. Robotic synthesis and assay platforms are genuinely expensive to build and to maintain. This is not software you spin up in the cloud. Second, chemistry generality. Automated synthesis works well for some reaction classes and poorly for others, and a lot of medicinal chemistry still stubbornly resists full automation. And third, the sharpest point from Episode 5, in full force, these labs accelerate discovery, they don't independently do it. Closed-loop labs have not discovered a validated drug end-to-end without human-directed biology guiding the target selection, the experimental design, and the interpretation. The loop optimizes brilliantly within a problem that humans framed. It does not yet frame the problem. So here's the framing worth landing. Self-driving labs are best understood as a throughput technology, not an autonomy technology, which is the same lesson as the rest of the series. Remember Episode 1, the value isn't that the AI is smarter, it's throughput and triage. These labs widen the validation funnel that everything upstream pours into. Picture the loop, robot arm, neural net, molecule out, result back in, the model getting visibly sharper each cycle, and then freeze it on the one node that's still a human, the person who chose the target, framed the question, and read the meaning. The automation is real and valuable. And it is genuinely not the same thing as AI discovering drugs all by itself.